Imagine a language written with only 4 letters. This language is DNA, the blueprint of life. It dictates everything about us. Frederick Sanger made it possible to read this language in 1977. His Sanger sequencing revolutionized biology. Like the printing press, it democratized knowledge. This earned him a second Nobel Prize in 1980. It paved the way for the Human Genome Project. Sanger sequencing opened a new era of biological understanding. It's a story of human ingenuity and the quest to unravel life's mysteries. So, how does this elegant method actually work? Picture a microscopic construction project where we're trying to replicate a single strand of DNA. We start with a single strand of DNA, the target we want to sequence. This DNA is mixed with a cocktail of ingredients, including nucleotides labeled A, T, C, and G. Nucleotides come in 2 forms, regular ones called dNTPs and special ones called ddNTPs. The special ddNTPs stop the DNA building process. An enzyme called DNA polymerase adds nucleotides, creating a complementary strand. When a ddNTP is added, the building process stops. This generates DNA fragments of varying lengths. Each fragment ends with a unique ddNTP. These fragments are key to deciphering the DNA sequence. They hold the secret code we're trying to crack. The next step involves separating these fragments based on their size. Now that we have our collection of DNA fragments, each tagged with a fluorescent dye, it's time to bring them into the spotlight. This is where capillary electrophoresis comes into play, a technique that separates the fragments based on their size, much like a molecular racetrack. As these fragments race through the capillary, a laser illuminates them, causing the attached dyes to fluoresce. Imagine a parade of colorful lights, each color representing a specific nucleotide. This dazzling light show is captured by a detector which records the color and order of the passing fragments. The detector then generates a chromatogram, a graph showing the color peaks corresponding to each nucleotide in the sequence. Picture a barcode, but instead of black and white stripes, we have a vibrant display of fluorescent peaks. This chromatogram is the Rosetta Stone that allows us to read the DNA sequence. By analyzing the order of the colored peaks, we can piece together the precise sequence of nucleotides in the original DNA strand. From this sequence, we can glean a wealth of information about the organism it came from. Section 4. Sanger's strengths, accuracy, and accessibility. Sanger sequencing is celebrated for its high accuracy and reliability, especially when reading shorter stretches of DNA. It's like a trusted magnifying glass, allowing us to zoom in on specific genes or regions of interest with remarkable precision. This makes it invaluable for a wide range of applications, from diagnosing genetic disorders to identifying pathogens. Moreover, compared to newer sequencing technologies, Sanger sequencing is relatively inexpensive and easy to implement, especially for smaller-scale projects. This accessibility has made it a workhorse in research labs and clinical settings worldwide, democratizing access to genetic information. Imagine a doctor being able to quickly and affordably diagnose a patient's genetic condition, allowing for timely and personalized treatment, or a researcher tracking the spread of a virus in real time, informing public health interventions. These are just a few examples of how Sanger sequencing continues to make a tangible difference in our world. Section 5, limitations on the ladder, throughput and resolution. While Sanger sequencing has been a cornerstone of genomics, it's important to acknowledge its limitations. One major drawback is its relatively low throughput, meaning it can't handle the massive amounts of DNA required for sequencing entire genomes quickly and efficiently. Think of it like trying to read a whole library book, 1 page at a time. Another limitation is its resolution, which makes it challenging to decipher highly repetitive DNA sequences. These repetitive regions can confound the sequencing process, making it difficult to accurately determine the order of nucleotides. It's like trying to read a sentence where the same word is repeated over and over again. Despite these limitations, Sanger sequencing remains a valuable tool, especially when accuracy and cost-effectiveness are paramount. It's like a trusty microscope, ideal for examining individual cells or small objects, even though it may not be suitable for studying the entire universe. Section 6. Sanger's legacy, a foundation for genomic exploration. As we venture deeper into the 21st century, the landscape of genomics continues to evolve at an astonishing pace. Newer, high-throughput sequencing technologies have emerged capable of decoding genomes at breathtaking speeds. Yet Sanger sequencing remains an important tool in the genomic toolbox. Its legacy extends far beyond its technical specifications. It ignited a revolution in biology, empowering us to explore the intricate workings of life at the molecular level. From personalized medicine to forensic science, from evolutionary biology to agriculture, its impact is felt across countless fields. Sanger sequencing serves as a powerful reminder that every scientific breakthrough, no matter how small, has the potential to unlock extraordinary discoveries. As we continue to explore the vast universe within our cells, we stand on the shoulders of giants like Frederick Sanger, whose pioneering work illuminated the path towards a deeper understanding of life itself.